once a year, I think.
Well, I didn't know that. I didn't know.
I didn't either. I wasn't there also.
Well, because now I can't even do that Thursday.
Because that's something people can make.
Yeah.
Not June.
Tuesday or Monday.
Yeah, literally. Ollie said that last week, so I was like, no one can make those. Like, it's not reasonable. And then she's like, well, a lot of people can come. That's why people shut up like 6.
Exactly.
And I'm like, oh, sorry. He's like, why are you walking so close to me? It's just whole thing. I'll be fine by keeping trust. 1 week. He said 1 week you'll be off. So. She helped me one time when I didn't take it. I'm waiting for the key so people can stand up and take this. I know, what can I say?
Did you make
No, they're not good.
I was coming out.
Oh, you just like QR code and we send the real I don't know that is from so let me give you I know. I was like cooking this and I spent 3 hours and came doing all this. Yeah, but then I'm like if I don't hear anything, do you want to go? It's learning up. I don't think
But then he left her. Oh, right. That's what it was.
Alright, so I think I sure girls. I know. And that was Martin, but like now it's only 1 p.m. on Friday. I think I'll probably film next Tuesday.
It's just not that. I don't think it's broken.
No, no.
I don't think I need to like, memorize everything down here.
Yeah.
You should have sent her that.
I combined mine all, I combined mine all in one thing.
That was mine. That's
So I don't have it, like, I have to.
Yeah. And even if you just like did something with like these two or something, like adding
I'm just typing on, I'm like, hand mine off.
No.
I'm gonna see my notes again.
You can take pictures of them. Right here.
I'm trying minimal. This was before. This is my.
Well, happy Friday, everyone.
Happy Friday.
Friday is my favorite day of the week. I don't know about you, but I suspect it may be for you as well. So, I begin with just a reminder that our Pre-Health Professional Society is hosting a meeting on Tuesday, the day after Martin Luther King Jr. Day, on Tuesday at noon. In the Peelle200, the Gurdon boardroom, and we're going to host the speaker from the Gift of Life, Michigan, and she's going to be talking about their organ tissue donor registry program. Now, later in this course, later in the semester, we're going to be talking about our immune system and how we have antibodies that recognize foreign cells that make it difficult for tissue and organ transplantation. So the speaker is going to be talking about the importance of having such a registry because of those difficulties with our immunological recognition. So, because it's related to our microbiology course, extra credit for attending on Tuesday. And I realized that not everyone has an opening on Tuesday at noon. So I'm going to try and record it for those who can't attend. But for those who do, or if I can get the recording to work, five extra credit points for you. I know you're thinking, 5, what does that mean? Well, 5 is more than 4. Okay, here's what it means. When we take our exams, those exams are out of 100 points. So our future exams will be out of 100 points. So 5 is half a grade. 5 is equal to half a grade. So if you score, for example, with a high C on an exam, with the 5 points extra credit, that will move you up to a B. Does that make sense? Kind of put things into perspective. Yeah. Okay, so remember we're not meeting Monday because of Martin Luther King Jr. Day. So this is your reminder. Okay. 5 extra credit points for attending on Tuesday or watching the recording.
Where's the meeting again?
Yeah, it's going to be Peelle200. So it's the first room after you enter the building from, what's that street called? Williams. Thank you. From Williams Street. It's called the Gurdon boardroom. So. Okay. All right, good question. Other questions?
What if we show up class Monday? We extra 10 points.
Yeah, I will not be here on Monday, and I don't think you will be either. Okay, no extra credit for showing up on Monday. As a reminder, we will be holding our elections for the officers for our Free Health Professionals Society on Tuesday as well. So thank you to all, for those of you who signed up. So your name will be on the ballot, and then if others want to sign up, just let me know after class. Okay, so let's do a quick review. So last time, what are some of the topics that you learned last time?
What are some of the commercial uses for microorganisms?
Excellent. So a lot of times we think of microorganisms as being harmful, causing diseases, but there are actually some benefits, right? Including practical uses such as, anyone? What?
It was used to clean up oil spills.
Yeah, clean up oil spills.
Make ethanol.
Making alcohol? Are you thinking drugs? No, I'm just kidding. Yes, fermentation. Yeah.
Food production too.
Food production. Yeah. Do you like your cheeses and yogurt and stuff like that? Yeah. Okay, good. All right. So what else did you learn last time?
How like diseases have gone down through vaccination.
Very good. So thankfully, through the modern science, the incidence of serious diseases have declined over the years due to a number of reasons, including vaccination. And what are some other reasons? Improved sanitation. How about one more?
Antibiotics.
Antibiotics. Yep. Drugs. More drugs. Okay. All right. Let me think. What else are we talking about?
More people died of that flu in the 1900s than in the World Wars in Vietnam and Korea and.
Yes, I mean, it's really tragic to think about all those who have, perished because of these diseases. And so I think that kind of stresses the importance of learning, education, and being able to, prevent microorganisms, some of these harmful microorganisms from propagating and preventing their spread. So that's going to be kind of a constant theme as we learn the materials from this textbook. So, okay, I think that was a good review. So let's learn new stuff. Or some of the stuff is probably old stuff, right, that you've learned before that we are reviewing. So that includes how scientists had a system for assigning a single Latin two-part name to every known living organism on the planet. Okay, so that's scientific name is different from a common name. Okay, how so? Okay, so let me explain something. When I go like this, that means you talk.
The common names aren't in Latin.
They're not in Latin, they're in the common vernacular, right, of the population. Yep.
Multiple animals can have the same common name. Multiple species can have the same common name.
Yes, there's confusion that can result, right? Because of you can have a species with, that share common names that are actually different. Okay. So think of the scientific names as a way of trying to kind of avoid confusion as you move from one population to the next. For example, I did a short a trip to a country in Africa called Uganda, and I did some mission work there, and they had some really beautiful vegetation there. So there's these beautiful trees that have red flowers on them, and I asked one of the locals there, what's the name of that tree that has the red flowers on it? And he told me, we call that the red flower tree. Well, to me, that really didn't mean anything. Like, okay, that's fine. And I said, well, what about that tree over there that has all those white flowers on it? And he said, we call that the white flower tree. So to me, I had no idea what we're even looking at, because that's the common name. I guess I should have asked him, what's the scientific name of that one? That would have been more beneficial to me. Okay, so we're looking at a two-word name, two-part name. And I know that may seem odd. Why do we have a two-part name? Well, think about your own name, right? Don't you have a first name and a last name? That's a two-part name. So if you think of it that way, it's not that odd after all. So why not give all species on the planet a two-part name like you? So the first part isn't, well, we don't call it the first name. Instead, we call it the genus. Okay? And then the second part here is what's called the specific epithet, also known as species name. And then collectively, that's the scientific name. And then we are using Latin because that's the traditional language of learning. And you probably have noticed this is in italics. Can you tell that? Italics, uppercase, lowercase, right? Uppercase, lowercase. E is an abbreviation for a much longer name. E is for Escherichia. Escherichia. It's no wonder they go with the letter E, because Escherichia has a really long name. It'd be hard to write that all out, right? Okay, so let's keep moving here. Oh, well, wait, I just told you all scientific names have two parts, but wait, there's a third part. There's an optional third part. And that's what we call with bacteria, it's the strain designation. So let's think about with people. Have you ever encountered anyone that has three parts to their name? Yeah, I have. Sometimes we have like hyphenated names, right? So there's two parts hyphenated together. Sometimes we have like a senior or a junior. Sometimes I've encountered people that have like a first or second after their last name. So there's multiple parts there. And then in some parts of the world, I'm thinking maybe like in Spain or parts of South America, there's more than just a first and last name, like the tradition in Europe and also in the United States is, for people when they due to marriage, sometimes often women will adopt the last name of their husbands, but in other parts of the world they don't do that. They just add their husband's last name to their own last name without the hyphen. And then you do that for multiple generations. And then, the names just keep getting longer and longer. Okay, so you can have many parts to a name. So, all right. So we're using the strain designation to distinguish one kind of E. coli that may be different from another E. coli, that are the same species, but they're a little bit different. Okay. All right. Why are we using microscopes in the lab? Well, that's because these organisms are very small, and they're not visible with your eye, right? So this, you may have seen this kind of pictogram before. Have you seen this one before in other courses? That kind of puts, scaling the sizes of the different kind of organisms. Okay, use your eagle eyes and tell me what size is our most bacteria? It's about 5 micrometers. That's right. So right in that range, right? Well, I guess there was a bracket there, right? But yes, very good. So the micrometer is also known as a micron. Okay, so those are synonyms for each other. 10 to the -6 of a meter, each micron. So in other words, think of maybe in your intro science courses, you played with meter sticks. And then the idea is if you take that meter stick, divide it into 1 million equal parts, each of those parts would be equivalent to a micrometer or a micron. So that's how small we're talking about. 1/1,000,000th of a meter. Okay. All right, there we go. Okay, we're gonna don't worry about recording all of this, but this kind of gives you an idea of what we're gonna be looking at and studying in the future here. So next slide, we're gonna look at different shapes. Chapter 3, we're gonna be looking at cell wall flagella. So don't worry about recording all this, but it is coming up. So let's review shapes here. All right, think back to Tuesday in the lab. What 3 shapes did you look at? Coccus, bacillus, Vibrio. Okay, very good, very good. So just as a reminder, coccus is the one that looked like tiny little dots. They're actually spheres or balls. Think of it as like a beach ball, but so small that they only look like a little dot, even under the microscope. Okay, so that dot, each dot was an individual cell. And then you also looked at some that looked like rods, and that was bacillus. So, and what we mean by a rod is kind of like my water bottle here. So just the blue part is kind of shaped like a pill. That's what we mean by a rod shape. Okay, so under the microscope, because they're so small, it just looks like a little line. But it's actually shaped like this. It's 3-dimensional. But that is only a single cell. And then Vibrio, looking at your keyboard, look at the shape of a comma. How it's kind of curved a little bit. That's Vibrio. Okay, then we have 3 more on our list here. Spirillum and spirochete. Now, under the microscope, they look the same. They're gonna look like little squiggly lines. Like, I guess they call them spirals here. And the difference between spirillum and spirochete is that the spirillum would be a fixed shaped spiral. So going back to, thinking about drugs, think about the corkscrew for a wine bottle. A corkscrew, it's fixed in shape, it's spiral. Okay, but then for the spirochete, that one's also spiral shaped, but that one has a flexible cell wall. So think snake, snake-like, where it would kind of wiggle back and forth as it moves it through the water. Okay, so it's not fixed. But under the microscope, they look kind of the same. Well, it's not the same. And then the last one's kind of interesting. Pleomorphic. These are cells that don't have a wall around them, but they do have a flexible membrane. So they're kind of like a blob. They can change their shape. They are a shape shifter. Have you heard of that term before? Yes. So there are bacteria that can change their shapes. Okay, let's keep going. Oh, pictures! Picture time! Okay, I can't quiz you because the words are actually on the slide, but at least you can kind of see, these pictures were not taken with a light microscope. Anyone know what kind of microscope was used here? Electron microscope. Good. Boy, I'm getting old. I can't. It's hard for me to hear what you're saying. But very good. Electrons, so even more powerful to get more up close pictures, right? All right, we're learning that these cells or that bacteria are unicellular, but for the most part. But what we learn on this slide is that those cells can actually adhere or attach to each other to form larger aggregations. So similar to the way that our cells are often joined together to form tissues. In this case, these are not tissues, but they form what they call arrangements. We'll see pictures on the next slide here. But the reason why they form these arrangements is because of how they divide. So it's because of the orientation of cell division. So in the first case, you could have the orientation of cell division resulting in a long chain of connected cells. So think about like beads on a string, for example. And we often see that with bacterium with the genus Streptococcus. So maybe some of you already may have heard of Streptococcus because this is what is the causal agent for strep throat, throat infections. The next one is sometimes we have arrangements in these clusters called packets. And then the last one is if the packets look like grapes. And we often see that with Staphylococcus. Okay. Okay, so you're probably wondering, what does a grape cluster look like? So think about, going to the grocery store, picking up a stem with grapes still attached to them, and that's pretty much what we're talking about there for the grape-like cluster there. So let's look at some pictures here. Okay, so first one, long chains. See, these are the chains. Okay, here's our packet. And then below that, the grape cluster. Is it making you hungry? A little bit. I don't know about you, but I'm always hungry. All right, so why are we learning this? That's because I mentioned on Tuesday we're going to be isolating bacteria from your body and from the environment. And we're going to be looking at them under the microscope. And we'll be able to see some of these different kind of arrangements, and then, based on those arrangements, kind of give us an idea of what they may be. So if we see something like this, it may be Staphylococcus. Recall that we have these different domains. So what are the three? Well, one's right up there. What are the other two? Bacteria and eukarya, for the eukaryotes. That's right. So can anyone recall? Okay, I was going to ask you if this was a eukaryote or prokaryote, but it's on the slide. Everything is on the slide already. This is a prokaryote, just like the bacteria. But how do they differ from bacteria? They have cell walls, but they're made up of different materials. So they don't contain the substance we call peptidoglycan, which we will actually learn more about in chapter number 3. So the cell walls are different. And then the ribosomal RNAs are also a little bit different too. So I know we reminded ourselves last time, but let's do it again this time. What's the function of ribosomes? Protein synthesis. That's right. So these cells can make proteins, but the ribosomes are going to be a little bit different. And then look at this one, extremophiles. You thought you were the only one that likes extreme things, right? But no, there's bacteria that like extreme things too. How about living in boiling water? Yeah, who's up for that? How about living in salt water? Yeah, I'm up for that. Okay, so has anyone ever traveled out west in Utah? What's the name of that big lake?
Salt.
The Great Salt Lake, yeah. So there's so much salt in there, most organisms would die in it.
But don't they say you can't drown in it because you're so buoyant because of the salt content?
Because you would float. That's right. Yeah, and I mean, there's bacteria in these thermal vents at the bottom of the ocean. I mean, it's crazy, really crazy. So that's where you'll find some of these archaea. And then this is a reminder that microorganisms are small, but they're not only bacteria. They can include fungi. So that includes yeast, that's used for baking. That's unicellular, but also multicellular molds. molds, you know, like things that are, you see with food spoilage, mildew, that slimy stuff that's in your shower. those are fungi. Microorganisms include photosynthetic algae, you know, like with the greens, the browns. I think there's like a golden or yellow algae out there. Okay, and then we have protozoa. So those are single cell eukaryotes. So think amoeba. And then the helminths. So those are things like tapeworms, hookworms, the other worm there, roundworms. Some of you have pets. Oh yeah, most of you have pets, right? So you may have, I suspect you have already been exposed to some of these worms and you didn't really know it because their pet, you know, your pets can pass them out through their feces. Okay, so one way or another, knowingly or unknowingly, you've probably encountered many of these organisms too. And we're going to actually end with, well, actually we're going to just look at some pictures here. So don't worry about, you know, writing all this information down here. So we're just going to see some pictures. So pictures of fungi. So we have I believe this is yeast over here on the left. Okay. And then we have a mold on the right. And then these little circles here, they're labeled conidia. Those are actually spores. So that's how the fungi can spread, you know, reproduce by spreading in the air. Next slide shows us an example of the algae. So you can see like chloroplasts, the green photosynthetic organelle within each of these rectangular cells. Here's our example of a protozoan. So again, one single cell here, filled with organelles. And then no pictures of the helminths, unfortunately. All right, we have time. We do have time, so we're going to actually move on to our next chapter here. So we're actually going to skip chapter number 2, which I remember correctly is chemistry. And we're actually going to move on to chapter number 3, which is about parts of the, like a typical bacterial cell. I mentioned last time I did create that assignment on Blackboard where you can upload your notes for the chapters. I did set up so you could have unlimited uploads, unlimited submissions. So in other words, you want to turn something in now and then turn in something better later, that's acceptable. You know, you don't lose any points or anything like that. Deadline is February 2nd. So you got plenty of time, but let me know if you're having any problems with that because I'm pretty new to, you know, I haven't used Blackboard in like 10 or 15 years, so you know, I may not have set that one up correctly. But let me know, and then I can fix it if it doesn't work. All right, any questions about that? About chapter number 1? Looks like you are taking notes, so that's a good thing. All right, let me see if I can find my my notes for my slides for the next chapter here. So let's see. Let's close that one out. And there they are. All right, so chapter number 3, where here's our our outline here. So we're focusing on parts of the cell. Now some of these will hopefully be a review for you as well, because a lot of times when you learn about cells, you're presented with pictures of eukaryotic cells, like an animal. Here's a typical animal cell. And here's a typical plant cell, and then they usually follow that up with, here's a typical bacterial cell. Does that sound kind of familiar with you? Anyone? Do they do that in your biology 131? Yes? Okay, cool. Okay. So if you were to dissect a bacterium, this is kind of what it would look like. I kind of like these cartoon pictures. But if you like realism, we have the electron micrograph on the right. So I don't know about you, but I like color, so I prefer the one on the left. So remember we learned about different shapes? What shape is this bacterium? Rod shape. What's another name for rod? Bacillus, that's right. This is our bacillus. Okay. And we're going to start off by learning a little bit about this brownish colored membrane, cytoplasmic membrane. Then we're going to learn about the blue colored cell wall, and then the capsule, and then after that, flagellum. And I'm looking for a label. Did they label? Okay, oh yes, they have the label at the very top. I can't reach it, but I'll try and point to it at the very top. Those little kind of yellowish colored, it's kind of like spaghetti. Peelle207. We have our Peelle207 at the. So that's the order in which the slides are going to be going in, and then we're going to delve on the inside. So let's start with the membrane. All right, so we're looking at the structure that defines the outer limits of the cell. So the contents of the cell are retained within and separates it from what's on the outside of the cell. You remember seeing pictures like this one before? That's our fluid mosaic model of what we believe the plasma membrane looks like. Okay, 2 layers, right? Here's 1 layer on the top, 1 layer on the bottom. So it's a bi-layer. Those are 2 layers, right? It's called a phospholipid bilayer because the majority of what you see here with the brownish colored circle and the little yellow squiggles below it, those are called phospholipids. And then this is a mosaic because there's a mixture of things here. We have the phospholipids plus we have proteins. And it supposedly has a consistency similar to oil. Think like vegetable oil, for example. Okay. All right. There we have it. All right, so what's on the outside of this the cytoplasmic membrane? Can you remember? Moving outside? Outside of that, like what's on? What would be that blue colored structure? It's a cell wall. That's right, cell wall. So hopefully we have the slides in. Okay, before we go to the cell wall, let's learn a little bit more about the membrane. Okay, selectively permeable. Anyone remember what do we mean by that? Selectively permeable. Yeah.
It lets in, I think it's small, is it polar molecules and, like the larger molecules, like it lets in specific things.
That's right. So selectively means some things can pass through, but other things cannot. All right. So we have some examples down here showing that, for example, water can pass through. Larger molecules like an amino acid or a sugar cannot. So things can be, so think of this as like a barrier that separates the inside from the outside. Maybe kind of like a barrier, but then the idea is that some things can pass through these pores, which are composed, in this case, of proteins. So there are protein pores that allow some things to pass through. Okay. Okay, now we're going to learn about the wall. Only two kinds, only two types. And they were named based on how they stained with some chemical substances that were actually developed by a person with the last name of Graham. So this chemical stain was named after Graham, and so some bacteria stain well, positively, so they called those bacteria gram-positive. Others did not, so they termed that gram-negative. So we'll see some pictures here in a moment of what the color differences are between gram-positive and gram-negative bacteria. But the staining differences are due to differences in the cell wall. Hey, there's that word again, the peptidoglycan. Okay, I stumbled with that one. Let me hear you say that one. Are you ready? Are you ready to say peptidoglycan? On the count of 3? 1, 2, 3. Peptidoglycan. Okay, there you go. Yeah, I actually struggle with some of these words because I was actually born with a speech impairment, so some of these things, it's tough. It's really tough for me. So what's this peptidoglycan composed of? Well, it's made up of these two types of sugars. So those are the glycans. One is N-acetylmuramic acid, abbreviated as NAM for short, and the other one is called N-acetylglucosamine, or NAG for short. And if you can kind of look at these colored circles of purple and blue, they're trying to show you that they're alternating. So you have NAM followed by NAG, followed by more NAM and more NAG. So they're alternating. So with those two kind of glycans or sugars, you can make long chains. So we have one long chain here, another long chain down below, and then we have a tetrapeptide. There's actually 3 of them here, connecting them together. So that's what they call a cross link. And then instead of just having 2 separate chains, try to imagine a whole bunch of chains all cross-linked together. So collectively, that's our peptidoglycan. Okay, so both gram-positive and our gram-negative bacteria have this substance. Okay, so here's our final 2 slides for today. Okay, time to wake up. We're almost at the end. Okay, so this second to last slide shows gram-positive cell wall, how that's all arranged. So on the bottom is our cytoplasmic membrane. So like the inside of the cell it would be below. So then looking up is on the outside. So what we see is a very thick peptidoglycan layer above our cell membrane with a small space called the periplasm. So here's our wall, here's our membrane. So if you kind of think back to the original picture of the bacterium, the blue cell wall is shown here, right there. Okay, now then you're all wondering, I want to see that last slide. It shows the gram-negative cell wall. I want to see how that differs from the gram-positive cell wall. Okay, I hear you, so I'm going to show it to you. All right, here's our final slide for today. Here's our gram-negative cell wall. And do you see peptidoglycan? Yes, it's right there. It's right above the cell membrane, just like the what we saw before, but this one's a much thinner layer. And wait, there's a difference here from the other type of cell wall. Now we see an outer membrane. So we have not only a membrane down here, but we have an outer membrane that's above or outside of that thinner peptidoglycan layer. So that's our gram-negative cell wall. And next time we're going to learn why we see a color difference when gram stain is applied to these 2 different kinds of cell wall. I can't wait. All right, everyone. Remember, we're not meeting Monday. We're going to meet again Tuesday. All right? So have a great weekend.
I